Welcome to the knowledge room, the concept of licenses and biotechnology. This is a lecture where we are going to learn the DNA fingerprinting, how it evolves with time, and how useful the DNA fingerprinting strategy in the current time, both in forensic science as well as in identifying people during the natural disaster. 99.1% of the genome is same for the human population. So the remaining 0.9% of the genome is used in identifying the individuals. Human gene polymorphism. Polymorphism is having the different forms of the same gene. So substantial allele frequency differences among the population. Individual ancestry lower, associated standard error and the major precision. The ancestry of the individual persons. So there will be the less standard error and it increases the precision or the accuracy of the similarity of the genome polymorphism. The number of markers to analyze needs to be very high for making the human polymorphism as a standard in identifying the individuals. So there are the variable DNA sequence, those are the polymorphic markers to differentiate and to correlate individuals. That is the center of the studies, or that is the focus of this lecture. Lineage or the genetic markers. Any genetic information that is uniparentally transmitted without homologous recombination of the polymorphism. So during the transmission from the parent to the offspring, there is no recombination and there is no polymorphism due to the mutations. So they're exclusively the polymorphism is generated by mutations. So there is no mutation, no recombination. So those are the lineage or the genetic marker that transmits from the parent to offspring. So it can be maternal lineage, that is, so we know through the mitochondrial DNA sequencing. It can be paternal lineage, that is to the Y chromosome marker passed from the father to the son unchanged. As we can see it here, there should not be any mutations. Lineage marker, the genetic profile referred to as the haplotype rather than genotype because there is only a single allele for individual. So the lineage markers are the haplotype. Y analysis is mainly used to analyze male and the female mixtures after the sexual assault. For the mitochondrial DNA is a closed circular molecule consisting of the 16,569 nucleotide pairs that are analyzed to study the maternal lineage. Genetic identity testing, the pattern of the genetic material that is unique to almost every individual. That's why it is known as the fingerprinting. Small number of the sequence differences are used to distinguish all humans. That means this sequence is the unique feature of an individual. This is used for identity testing. So the technique that is used to identify individuals can be called as DNA fingerprinting, DNA profiling, or the DNA typing on the alternate fashion. They are interrelated, or it is the same technique with the different names. So the person who made or who initiated the DNA fingerprinting is the Sir Alec Jeffreys. He was a geneticist on the University of Leicester, UK, and he used the DNA based identifying individuals. He was working on the microglobin gene in the seal that is homologous to the human counterpart. And he found that the short repeating sequence which are homologous in nature. And these tandem repeats are called the mini satellites. Repeating sequence could be highly variable and informative genetic markers. So these tandem repeats or the mini satellites, these are highly variable or the polymorphic. It is having different or the unique, it provides the uniqueness to the individuals. And thus it is the informative genetic marker. How it is done? DNA has to be extracted. It has to be subject to the restriction endonucleotidization. And whatever the pattern of the DNA segment, we see almost 15 to the 20 bands per sample. Then it is, these bands are seen in the agar gel electrophoresis and finally the detection or the bending pattern. That may be through the autoradiography or through the precipitation technique or the luminescence technique. So this is the first paper of the 1985 related to the DNA fingerprinting, and by the end of the 1986, DNA fingerprinting gathers a lot of public support, and it is used as a very well established technique in identifying people. So the early days of the DNA fingerprinting goes back to a couple from the Ghana. They migrated to the United Kingdom. So this couple has 4 children. One of these children, he moved from the UK to the Ghana. So when he was returning, there was a problem in the identification of this one child. So it was very difficult at that time because the father was not alive at that moment, and this mother has 3 sisters in Ghana. So it is a very huge task to identify that this child is from this mother who is a UK resident and not the child of any of the sisters who are residing in the Ghana. So Geoffrey, he matched the DNA profile of this child with the mother as well as the 3 undisputed children, those who are living with the mother in the UK. And that this child belongs to this mother was established. Then the second case, a single locus probe recognizes at most 2 DNA segments in an individual corresponding to the 2 alleles, mother and the father. This was used in a double murder case in the 1986 by the same Jeffrey. So there are 2 15-year-old girls in Leicestershire had been raped in the murder. So the time period was 3 years difference, and it was considered as 2 different murder cases. And they found they were actually capture a different persons, and that person was in the custody. But after the work of the Jeffrey, it was identified that the 2 murder was done by one individual, and that is not the person that was in the case. So that person was released, and they find out they actually did the DNA fingerprinting of that entire area, and finally they find the persons. And after the DNA matching, they found that actually these 2 murder belongs to the person who committed the crime is only one. That is the one person, and they named him. Then there is a mini satellite that is also called the variable number of the tandem repeats. The unit length of the repeating unit is 6 to 100 bases. The units can be repeated 2 to several hundred times at each mini satellite. Cluster near the telomeres or the end of the chromosomes. This is the one molecular marker that is used for DNA profiling or the DNA fingerprinting. The another is the microsatellite, also called the short tandem repeat, that is very much popular. The units are 1 to 7, most probably it is 2 to 5, most commonly. And it is the repeats of 5 to 100 times in the microsatellite region. And this STR was used for a Nazi, Josef Mengele, who was known as the angel of the death, and to confirm his identity. And in 1979, he was thrown and buried in the Brazil. His body was recovered in 1985 from the Brazil. And after that DNA testing and the reverse paternity testing with his wife and the son in 1992, it was confirmed that that body they recovered from the Brazil belonged to the angel of the death, Josef Mengele. And that credit goes to the short and long repeats DNA fingerprinting tool. So where it is used, short and long repeats, paternity identification, guilt or the innocence, assessment of the donor hematopoietic engraftment after the bone marrow transplantation, chimerism analysis after the solid organ transplantation, specimen identity, malignancies of donor or the recipient origin, polymorphic characters of the polymorphic characters of the trait that is inherited in the Mendelian fashion. So this is one STR signature, this is the maternal, this is the two nucleotide repeats, 1, 2, 3, 4, 5, 6. So there are 12 nucleotides. You can see there are 12 nucleotides, here are the 12 base pairs. The paternal is 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 18 bases here. So this is the maternal allele, this is the paternal allele of this locus, allele of this locus of the STR. And this is the STR profile of the recipient and the donor before transplantation. And the asterisk, it indicates the informative allele at each locus. So this is the sex identifying STRs found in the X and the Y chromosome Amelogenin. And these are the STR D8S 1179, D21, S11, D18, S51. And this is the evaluation of the chimerism status of the STR analysis after the liver transplantation. So this is the recipient, there are two bands, this is the donor. So they do not match with each other, but when there is a chimera, you can see this band corresponds to the small band. Similarly, this band of the donor corresponds to the small bands. So we can evaluate the chimera by the DNA fingerprinting process, that is to the STR. Then restriction fragment length polymorphism, the samples can be from the blood, semen, bone, skin, cell, urine, feces, and the cells at the root of a hair follicle. Expose the DNA to a set of restriction enzyme is the next step after collection of the sample. Then no single well-defined in the predicted pattern the bands for the human genome. There is a characteristic pattern the bands for a given individual. So for each and every individual, there is a signature pattern, but there is not specific pattern for an human species. So these are the eco-R1 restriction site G8 etc. You can see the size of the target DNA can be 1000, 2500, 4500. But if there is a mutation in one of these restriction endonucleosides, so it cannot be recognized by the eco-R1 now. So instead of two bands here, there will be 6500, one single band will be seen. So here you see this is the 2500, this is the 4000, 2500, 4000. But when there is a mutations here, we don't see this two band, but we see this entire B band, that is the 6500. So if we only consider this three band, this is the 2500, we know the 6000. If we put a marker, the probe here in the 2500 area, so from this suspect one, you can see 2500 band, and from the second suspect, because it cannot be fragmented too, so we can see the bigger band 6500. So DNA fingerprinting, the RFLP needs more than 25 nanogram of the initial amount of the DNA, and it is to establish a link between the biological evidence and the suspect. So there is the DNA sample collected for the crime scenes, and there are the victims, suspect 1, suspect 2 officers. Means DNA fingerprinting is done. And after that, we can see that this is common everywhere, victim, and this is suspect 1. So these three, and this is common to the officers. So these three are moved. This is same everywhere, this is gone. Similarly, this 2 is only victim sample, so this is gone. These three belong to the victim, that's why these three are gone. Here this belongs to the victim, and this one also belongs to the victim, so this is gone. And in this one, suspect 1, we can see there is a band present here. So that is not present in the crime scene, so this person is not the criminal. but the suspect 2, all the bands present in the suspect 2 are present in the crime scene and they do not represent the victim sample, so suspect 2 is the criminal. So this is another example. So in the suspect 1, this is the suspect 2, all the means bands of the crime scene matches with the suspect 2. So in this case, the suspect 2 is the criminal. So the DNA fingerprinting got the wider acceptance, like identification of the last Russian jar from a collection of the bond bones using the RFLP technology. So it is the jar Nicholas 2 short tandem repeats and STRs are 2 to 5 base pairs DNA the repeats. Before we have seen it is 1 to 7. So it can be most probably 2 to 5, but the range can go up to the 7. So this is one of the STR. So this is the repeat of the 4 G8 and there are 1, 2, 3, 4, 5, 6, 7, 8. So this is the STR called the very well known STR D7 S820. What is it? D for the DNA, S is the number of the chromosome, S7 is the number of the chromosome, S for the single copy, 820 is the locus number. So it is the chromosome 7, as you can see here, and this is the 6 and the 11 gata repeats. So in one allele it is 6, in another allele it is 11 repeats. But a friend might have 9 and the 14 repeats. So that D7S 820 for the two individuals, the bending patterns is different. In one it is 611, in another it is 914. And the STR sequence can range from the 3 to 21 repeats. And D7S820, the range is 6 to 14 repeats. So this is the number of the repeats, 6 to 14. STR is that they are flanked by known sequence. So we know the sequence that is present on both borders of the STR. So we can design primers and we can amplify. So the quantity of the DNA is not the limiting in starting the STR. So the number of the repeats in STR is a finite number. There are a finite number of combinations for their length, like the 3 to 21, that is the number of the repeats, and that can yield the 190 combinations. How it can yield a 190 combination? So 3 to 21 means there are 21 minus 2, 19 combinations. 19. Okay, so the repeats are 2 to 5. So 2, 3, 4, 5. So there are the into 5. So this is for chromosome. So in the homologous chromosome, again there are the 2. So 19 into the 10 equal to 190 combinations are possible for D7S 820. And small probability to share the same pair of the STR length. So coming to the next one. So this is the number of the combinations of the allele. This is the 14 repeats maximum possible, minus 6 plus 1. This is the n minus r factorial. So this is the n minus r into n minus r plus 1 by 2 equal to 45 combinations of the allele lengths are there. So there are 6 to 14 repeats. Number of the combinations, so the n objects taken at 2 at a given time, this is the n factorial. So n into n plus 1 by 2. So 6 and 11 repeats would show up as equivalent to 11 and 6 repeats only there. Then for a specific DNA fingerprinting using the 13 similar STRs. So this is coming up to be 45. So 45 in to the power 13 because there are 13 STRs. So there is a possibility that the two individuals will have the same STR bending patterns when the population will be 3.1 into 10 to the power 21. But the planet Earth, where we are living, the population is 7.9 into 10 to the power 12.9, which is a very small compared to 10 to the power 21. So on this planet, when you consider the 13 STRs as identifying features of an individual, there is no possibility that two individuals having the same bending patterns. So STR is a very useful tool in identifying or giving an identity to an individual. And we know the sequence that is flanking the STR, so we can PCR amplify. So only 1 nanogram of the DNA is enough in doing the STR analysis. So these are the 13 core STR loci and Amelogenin. And as I discussed, D for the DNA, 3 is the chromosome number, S is the single copy, 1358 is the chromosome number, and this TPOF, this is based on the protein, FGA, TPOF, CSF, 1PO, these are all based on the protein. Their names are given. So CODIS is a combined DNA index system. It is maintained by the US Federal Bureau of Investigation. And in the US, according to the legal system, all the 13 agreed loci that is present here and the Amelogenin has to be matched in identifying an individual. In UKR, in the United Kingdom, instead of 13, it is a 10 STR loci that is used for the identification purpose. And it is called the National DNA Database or the NDNAD. And the system used in the European is called the European Standard Set ESS of STR. And this is the 23 in the various parts of the world. Those are used in identifying individuals. So this is the name of the STR. This is the chromosome location. This is the physical position. And this is the category and the repeat motif like the TAGA, AATE. So this is 4 nucleotide, 4 nucleotide. Most of them are the repeats of the 4 nucleotides. This is 3 nucleotides. And this is 5 nucleotides. So other than the STRs and the mini satellites, single nucleotide polymorphism, base substitution, insertion or deletion called the indel at single position in the genome of any organisms are used for that identifying individuals. And if there is any lacuna in the RFLP and the STRs or the mini satellites, those are filled by this type of the DNA, means chromosome properties. So SNP with respect to the STR markers increase the sensitivity of the test in the conditions like the severe fragmentation of the DNA. Then it is biallelic in nature. So it provides in a higher level of the multiplexing, so automation. Next is the indels or length polymorphism. So based on the length of the indel, it varies from the individual to the individual. That's why the length is the polymorphism. Then the range is 1 nucleotide to the hundreds of nucleotides. It can be tied by the capillary electrophoresis. It is biallelic, means in one individual you can see 2 types of the indel, and this is defined as the short interval allele length variation. Then there is a combining many of the desirable features of the STRs and the SNPs. Indel can bridge the gap between these established strategies. So STR, SNPs, and the indels, they will provide a robust information about the identity of an individual. So a multiplex of the 38 biallelic indel markers can recently been developed, making this kind of marker an important and efficient alternative to SNPs and the STRs, or they are complementary in nature. And the first government database was prepared in the UK. They use the 10 STRs as the agreed upon. Means all the 10 STR has to be matched in identifying in individual. There is a high throughput method for the DNA profiling or the DNA fingerprinting approach that is called lab on a chip. So in this case, there is a microfluidic genetic analysis system all sample processing steps in a single micro device to develop miniaturized platform that integrate all aspects of the analysis from the sample preparation to the nucleic acid trial. That is a fast, automated, and efficient methods of DNA profiling. Thank you.